Okay, so it's my pleasure to welcome today's speaker because this is somebody I've been trying to get to come to Circus for a very long time. So I first met Deb 3 years ago when she was, of course, doing something completely different, but she mentioned that she was responsible for data quality at the Dana-Farber. And I thought, now that sounds like a really interesting problem. So I'm assuming most of you in the room know that Dana-Farber is the Cancer Institute in Boston and has done pioneering work in a number of fields. And I thought, that sounds like a really interesting problem. But of course, since then, Deb has gone on to do amazing and wonderful other things and is now heading up cancer research across 7 of the partners hospitals, right?
Yeah, I'll explain.
Yeah. Okay, she'll give you more detail. But basically, when it comes to medical research and data and interesting applications, and then, oh, by the way, saving people's lives, this seems like a really good confluence. So it's my pleasure to introduce Deb Schrag, who is both professor at the Harvard Medical School as well as the Harvard School of Public Health, and just a really interesting person. So, take it away.
Thank you. So it's my pleasure to be here. What I am not is a computer scientist. But it's my privilege to be here today, and really what I want to talk about, and I hope do, is entice those of you who are students or postdocs or soon-to-be researchers or faculty. I want to entice you and excite you about the tremendous opportunities that there are in medicine in general and cancer medicine specifically. The thesis of my talk is we really need your skill sets and expertise, and I want to walk you through a few examples as to why that is. So let's try to get next slide. Let's see. Seems to be okay. So what I want to do today, and by the way, we don't have to get through all the slide sets. I'm happy to take questions. Please jump in and interject. I've tried to eliminate as much medical jargon as possible, but I'm very happy to take questions along the way. So what I want to talk about is briefly review the traditional model for developing cancer treatments, then talk about the evolution and the rationale for what we call rapid learning health systems, specifically in oncology. And then most of the talk, I'm going to focus on integrating PROs, which stands for patient-reported outcomes, into cancer care. Talk a little bit about the current raging war between precision cancer medicine believers and skeptics. And then if there's time, we'll get into a discussion about our challenges with respect to integration of so-called genomic data and phenomic data. So here's what I'm not going to discuss at all. I'm not going to discuss data security and privacy. The basic premise underlying this talk is that health data must be kept confidential and secure, and that technologies to accomplish this core task exist. They haven't been well implemented. They need to be expanded and developed. That's another problem for another day. This talk presupposes and presumes that those technical, regulatory, and legal challenges have been successfully addressed. The other thing that I'm not going to talk about today is data sharing. This is a huge, tremendous problem. It is probably a topic for another, not day, hour, but month or year. It's incredibly important. I'm going to summarize it by saying data sharing is a good idea. It needs to happen. We're supportive. So this talk really presents a lot of research that's taken place over the last decade when data sharing efforts were, I would say, in their infancy, but presupposes a vision where we'll have a lot more data sharing. So this is what the biomedical translational research paradigm looks like. We start with what we call, and this is our biomedical jargon, we talk about T1 research, where we translate scientific insights to humans. So that's research on molecules, viruses, bacteria, non-human organisms, and figure out how to translate that understanding and knowledge to humans. The next transition, T2 research, translates that knowledge to actually make an impact for patients through typically clinical trials. T3 research translates that knowledge to practice, and T4 research to population health. we make a very big deal about distinguishing between two core different kinds of research questions in cancer medicine. One is efficacy. Can a treatment work under experimental conditions? And effectiveness. Does a treatment work when it is actually scaled up, rolled out, and implemented in real-world settings? Those terms are often used as synonyms, but for us, efficacy and effectiveness are very different, and we think about those concepts as distinct. Most of cancer research is T1-2. Yep.
Just for those who aren't in the medical field, can you give an example of where that difference matters?
So where something works experimentally but just doesn't scale up to population level?
Absolutely. So I can unfortunately and sadly give you hundreds of examples. And this is one of the gigantic failures of, I would say, the biomedical enterprise. If I'm trying to develop a drug to cure colorectal cancer, I'm a colorectal cancer doctor. When I leave here, I'll be going to see my colorectal cancer patients. And colorectal cancer patients' tumors metastasize to their liver. So their livers are full of tumors. So, oh, guess what? The livers don't metabolize and clear the drug very well. When you do your clinical trials, do you take everyone? You don't take everyone. You say, you want to do good experiments. So you say, if you can't get out of bed, I don't want you. If you're pregnant, I don't want you. If you're over 80, I don't want you. If your liver's too full of tumor, I want you. If you've had major bleeding in the last 30 days, I don't want you. So clinical trials have lots and lots of restrictive eligibility criteria. It'd be like saying we only want Harvard students with, B+ and above averages, or something like that, and that we're going to develop sort of all our teaching and learning methods only on this very cherry-picked selective group. Then what happens? The drugs are FDA approved, and they are then unleashed on the population at large. So, this happens every day, all the time, across all of medicine. Just to give you an idea of how extreme this is, in colorectal cancer, the median age at diagnosis is 72. The median age for participants in clinical trials is 61. That means we're discovering how drugs work since 61, it's a range, but in younger people. And then are we surprised when there's different toxicities in older patients who take a lot more medications, have a lot more additional health problems? So yes, it can work. A cancer drug can work in a healthy, appropriately selected population. That doesn't mean that it will work or it does work when you unleash it. Those are patient factors. There's also doctor factors. Doctors may dilute the medication, they may not read the label, they may not administer it properly. The patients may require coming once a week, they may skip treatments and only come 2 or 3 times a week. The medication may need to be taken how many of you have taken antibiotics 4 times a day and consistently taken all the medication exactly the way you were supposed to? Okay, so you're a computer scientist, but most of the world doesn't do that. And so drugs, the theoretical, we call that sort of efficacy does not match effectiveness routinely. We call this construct in our jargon, that's the implementation gap. Okay, and it's multifactorial, and we spend a lot of time and we do a lot of research trying to unpack the implementation gap and figure out what its source is. Did I answer your question?
That's great, really great.
Okay, but hundreds of more examples. So what are the goals of cancer medicine? They're really basic and fundamental. We sometimes lose sight of them. It's important that we do not. They are to help people live longer and help people feel better. And the endpoints that we are able to measure in cancer clinical trials, which is how we build most of our knowledge, is we measure overall survival, the burden of symptoms, quality of life, and sometimes we have to use surrogate outcomes. Disease-free survival, which is the time that the cancer goes away and doesn't come back. So you have your breast cancer operated on and removed, how long you remain free of the disease until it recurs, or progression-free survival, you're on a pill, you're taking a medicine, You're living with your cancer. It's there, it's not gone, but it's not getting worse. So those are our core outcomes, if you will. Cancer drug development traditionally is long, intensive, and expensive. This is just an infographic about the phases of drug development. It takes a long time to develop a new cancer drug. There's a trope where we start with what's called phase 1 clinical trials, which just establish the toxicity, whether you can give the drug safely to humans. These are non-controlled small trials, typically with under 50 patients. We then move to phase 2 trials, which typically involve, actually in cancer medicine, often less than 100 patients. They're just checking to see if there's any signal. Does this drug melt these cancers at all? Does it do anything? And then finally, drugs that make it past all those hurdles move to sort of the holy grail, which are phase 3 randomized control trials. Lots of fall off along the way. So what does a drug need to make it all the way to being approved? Well, starting in around the 1920s, the FDA was developed, and from the beginning, it's clear that drugs need to be safe, consistently manufactured, and the product needs to be the manufacturing needs to be high quality. In 1962, the original standards were developed, and what they say is that a drug must have efficacy, not effectiveness, but efficacy, demonstrated in adequate and well-controlled investigations. The plural is key. To get an FDA drug approved, you have to have traditionally 2 independent investigations. In 1998, the FDA Modernization Act relaxed those criteria a little bit and said, okay, we'll approve a drug if you have a single randomized trial, but it has to be really big, really large, with a really compelling benefit, and you have to establish and prove to us that repetition of that experiment would be unethical. No patients would participate. Very recently, just 8 years ago, we had our first guidance on standards for integrating patient-reported outcomes into the drug approval process, but historically, patients are completely absent and not integrated or a part of this process at all. So where does this process leave us? Many people, you read so much about cancer medicines, people think that we have tons of cancer drugs. We do not. Colorectal cancer is the #2 killer of men and women in the United States, so prostate and breast lung is the most common, and prostate and breast are #2 for men and women respectively, so colorectal cancer is #3 or #2 if you look at men and women combined, and it affects around 140,000 Americans every year. We only have 10 FDA-approved drugs. That is a very small number of medications to treat one of the major US cancer killers, and those 10 drugs, some of them are copycat Me Too's, so we really only have 7 distinct approaches. I want to describe for you, because it relates to how we think about data acquisition and analyzing data, the story of how we learned about one of those drugs, and that drug is called cetuximab. Has anybody heard of it? Cetuximab, Erbitux, has anyone heard of it? Okay, bonus points. Who went to jail for insider trading based on the drug cetuximab or Erbitux? Who spent time in jail for insider trading that drug? Martha Stewart, exactly right. Very good, yep, she did. That's why she went to jail. So I have to say, I was part of the foundational clinical trials and I worked with a large group of students and postdocs, folks much like you, doing the clinical trials, looking at the data, analyzing the data. These are folks earning $40,000 a year, research assistants. Not one of them went out and conducted any sort of insider trading, although they all had access to the same information, in fact, much better information than Martha Stewart. So these 2 drugs, cetuximab and panitumumab, are cousins. They're Me Too competitors. Their mechanism is understood, and they really are part of this sort of wave of precision medicine drugs. They're antibodies that inhibit the epidermal growth factor receptor, and we really understand how they work as a result of very, very elegant science that's shown in this little cartoon. But essentially, they bind the epidermal growth factor receptor, and that turns off a cascade pathway that results in neoplasia. This is the evidence base that the FDA had to consider approval of cetuximab in February 2004. Martha Stewart's insider trading was 18 months earlier, and the FDA rejected cetuximab at that time because the data were too skimpy. This is the data that ultimately led to the drug's approval. Most people look at this data and say, wow, that is really skimpy data. Look at the numbers, okay? So multiple independent studies. There were 3. The last 2 shown at the bottom, 57 and 138 patients respectively with advanced colorectal cancer, so these patients were going to die. In the first study, 9% of them responded to the drug. The second study, 15% of them responded to the drug with 138 patients. And then the manufacturers, or the pharmaceutical sponsor, was really excited, okay? Most normal humans look at this data and say, this was exciting? I mean, this is pathetic, right? But they went on and conducted a randomized trial, and it's hard to do randomized trials because patients always want to get the new drug, so they did it 2-to-1, so twice as many patients got the investigational study, 218 versus 111. And 23% of the patients who got the new drug had a response. What does that mean? It just meant that their tumor shrank on CAT scans. Only 11% of patients who got the new drug alone. Was there a placebo control? Not at all. Is that what you would really want to see if you were designing the perfect study? Absolutely. So this is not even a perfect study. What was the benefit achieved? The time to progression was 4.1 months versus 1.5 months, but that met threshold criteria for statistical significance. What about the impact on median survival? 8.6 months versus 6.9 months. Is it worth it to take this medicine for an extra, it's basically 6.5 weeks of survival at the median? So this is a pretty small benefit. But this study was a randomized trial published in the New England Journal of Medicine in 2004, and indeed is the foundation on which the drug is FDA approved. This is what the survival curve looks like, and you can see that if you look out at about a year, those curves are superimposable and converge. So people talk about the power of precision medicine and these amazing medicines. It is amazing. It's amazing science, but often the benefits are very small. So the drug is approved in 2004, and we started using it, and the company made billions and billions of dollars. So somebody can look up on some stock market website just how many billions. It causes me too much pain to look at that, but they made a lot of money, and it was FDA approved, and people started using it around the world. 4.5 years later, in 2008, this paper was published based on the insight that the tumor doesn't work if patients have a particular mutation in a particular gene. So again, discovery of the drug was made possible because of insights into molecular biology and cell signaling and how monoclonal antibodies work. But then tumor sequencing of patients' individual tumors said, gee, about 30% of patients, even more, almost 40, have a KRAS mutation, and patients with KRAS mutations do not respond at all. It's a complete waste of time to treat these patients with this medication. How long did it take us to learn that? 4.5 years. How many, not hundreds, thousands of patients were treated in the meantime? Many. That is a failure of data analytics and computational power, and that is a challenge that I put out there for all of you. The fact that that took us 4.5 years to figure out is embarrassing. But it actually, these are the survival curves that basically show on the top, patients who had a KRAS mutation, with or without cetuximab, survival is identical. But patients who don't, wild type patients, there's a big difference. So as shown on the bottom, if you're wild type, you definitely want to get this drug. And if you're not, it's a complete waste of your time. Then more recently, just about 2 years ago, we learned that other mutations, less common, not in 30%, but that in 10%, don't work for colorectal cancer. Again, by doing molecular profiling, precision medicine, and figuring out who these drugs work for and who they don't. Then, just this past year, we learned something else that's embarrassingly simple. The colon is an organ that starts on the right side of the body, goes up and across the midline, and down on the left, right? You think in high school biology. Well, it turns out that we consider all colorectal cancers the same. But from a biological perspective, they're not all the same. Tumors on the right side of the colon are molecularly different. They have different molecular profiles and molecular signatures. And it turns out that cetuximab doesn't work very well at all for tumors on the right side of the colon. Again, we learned this in 2016 for a drug that was FDA approved that we've been using in patients since 2004, and we didn't figure it out sooner. So this is what the timeline looks like for learning about cetuximab. It was initially approved in 2004. We found that it doesn't work if you're KRAS mutant in 2008. It doesn't work if you have some other less common genes present, about 10% of the population in 2014. And in 2016, we learned that it doesn't work for tumors in the right colon. Everything after 2004 was learned from mining what we call, so either post hoc analysis of existing randomized trials or so-called real-world data from treating patients in the real world. When the drug was approved and we gave it to patients, no more than a quarter experienced any kind of benefit. And now, as a result of this progress, when we give the drug, it works in more than 50%. So the core challenge that we face is how to accelerate this learning so it doesn't take 12 years. That's a general overview of precision medicine and why we really need to accelerate rapid learning. Because 12 years, I mean, this is embarrassing. And if this were just swallowing a Tylenol that doesn't really do anything, it would be really not such a big deal. But this is what it looks like to take cetuximab. In fact, the people who respond to this drug get terrible acne. We have 75-year-olds with terrible acne. This is what it looks like. And in fact, when people don't get the rash, it actually means that they will not respond, and we've actually learned that it's possible to stop the drug. If people don't have a rash 1 month in, the drug is a waste of time. Somebody had a question?
Yeah, so were you able to go back to the earlier data now and take out people who had these mutations or who had the wrong-sided colon cancer and then see what the survival was of the population that really should have been your target?
Yes, exactly. And it's much higher,
Yeah.
In fact, that is exactly how those studies were done, by going back and reanalyzing the foundational clinical trials. But you saw the foundational clinical trials as such skimpy data. It's just a couple of hundred patients in a disease that kills 50,000 Americans a year, and we can't get, I mean, worldwide it's huge. I mean, the studies are also international. So we have very few patients just because of how much, it's getting people to go in clinical trials, conducting them, executing them is challenging. So yes, that's exactly how it was done. Make sense? Yep.
So, given what you've learned in the last 12 years and looking at the curves, is the intuition that there is still progress to be made in understanding how the ones that don't fall into the categories currently combined actually do in fact self-identify and fall into different classes?
Yes. So if you take the vision of precision medicine to the extreme, right? So we start out and we were excited about drugs that 10, 20% of people would respond to, right? Now we're getting up to 50%, 60%, but that still means you're treating a lot of people who are getting no benefit. So the ultimate goal of precision medicine is we give no one a drug that they're not going to respond to, right? That's the ultimate goal. Do we have, we are nowhere close to a ceiling effect. And some of that so is there more discovery? Yes. A lot of the low-hanging fruit has been picked off, right? And now it's down to sort of complicated interactions, germline, somatic, tumor profiling. So it's interactions with other drugs patients may be taking, right? So there's certainly more to be mined and learned here. So this is what the FDA label looked like for cetuximab in 2004. And this is what doctors told their patients, and this is what patients read on the instructions they got from their pharmacist. You know, you go at Walgreens, they hand you that piece of paper in nano print that nobody ever really reads. But if anyone reads it, it's cancer patients because the side effects are really pretty terrible. And you know, this says, if you look in the second paragraph, that 88% of patients get an acne rash, and that 73% experience real fatigue. So this is not really a super fun drug. So you want it to work. So what we have historically been terrible at in cancer medicine is integrating the patient voice and learning from the patient experience. All these trials are done with the patient as a passive actor, not integrated, not involved in data gathering, not as an active participant at all. And I'd suggest to you that we've really done that at our peril. So I'm going to give you an example, sort of an N of one story. In about 2002, I was treating a patient on the phase 2 clinical trial that shown in the earlier data, that led to the approval. And I had a young man, a tennis player who was actually he was a computational scientist, and he was writing advertising algorithms for Google at the time, very early in 2002, and he had terrible fatigue. And he was dying, and he knew he was dying, and he went to the Berkshires for a weekend, and I got called by the Berkshires local hospital that this 32-year-old man could not sit up. He was so exhausted. So it turns out that at the major cancer center I worked for, we didn't figure it out, but they checked him for calcium. And his calcium level, calcium's an essential electrolyte that we all need for our nerves to fire and to maintain normal homeostasis, and it was undetectable. Well, low calcium is really an unusual condition, but one of the things that causes it is low magnesium. He was given oral magnesium, he didn't get better, he still couldn't sit up. We never monitor magnesium. It's just we monitor people's blood counts, but we don't check magnesium. No one had checked magnesium. The people who developed the drug hadn't checked magnesium. So at this little hospital in the Berkshires, I was on the phone with them, it was late at night, I said, calcium that low, check the magnesium. Magnesium was low, we gave him intravenous magnesium, he got up, he got better, and he played tennis the next day. So it turns out we went to the physiologist, and the drug cetuximab, those EGFR receptors are expressed, again, reverse translation, are expressed in the loop of Henle. We figured out the physiologic mechanism, the drug blocked them. You can't hang on to or resorb calcium or magnesium. So people who take this drug, about 25% of them develop magnesium wasting, and that's why 75% of them have terrible fatigue and can't sit up. So again, it took us a while. I figured this out in 2005, August. It's published in the Journal of the National Cancer Institute. What do you think happened when I went to try to present it? I'm not the drug company. They were not happy with me. Because this little discovery, what does it mean for them and their drug? A label change, a warning. It has to go out to people, right? Because we're hurting people by giving them a drug when if all we do is check a magnesium level and give them not oral, because the kidney is broken kidneys, the mechanism, and give it to them intravenously, we can fix the terrible side effect. But what's the mechanism of discovery of that? It's the traditional mechanism. It's no different than John Snow and the Broad Street Pump. It's just awake physicians or serendipity. It's not a system. And as medicine has grown more complicated, we need systems, and that's why we need computational power to figure this out in a much less haphazard, random, serendipitous way. So I'm pleased to say that although the drug company was not very happy with me and worked really hard to hope that this would go away, this was validated, and it's now on the drug label and has been there ever since. And patients come in for their magnesium infusions and do better with the drug. But that core insight that we lacked a way to take information from patients. Remember, hundreds of patients had been getting these drugs for a year and a half, complaining of terrible fatigue. We knew it, but we didn't do anything about it. We just let it be. We didn't gather the data, we didn't link it, we didn't integrate it, and then we didn't act on it. So, together with a number of my colleagues, we got really interested in integrating so-called patient-reported outcome or patient-reported data into clinical research. So what are patient-reported outcomes? They're basically any report on the status of a patient's health condition that comes directly from the patient without filtering or interpretation by another party. PRO instruments can capture a whole range of concepts that are important to patients, including symptom changes, levels of functioning, health-related quality of life, satisfaction, and treatment adherence. So, what's the motivation for measuring patient-reported outcomes? Symptoms are really common in advanced cancer and other serious illness. They interfere with people's activities. They put people in the hospital and send them to the emergency room and drive up healthcare costs. They are not systematically elicited, and even when they are elicited, and what I mean by systematically, doctors often say, how are you doing? How'd you do with your treatment? That's an elicitation, but is it systematic? Does it get tracked and recorded? Not so much. So this leads to huge missed opportunities to alleviate sufferings. So we have very entrenched systems that do not serve us well anymore. They did at one time, but they impede learning. In research trials, we're not total idiots. We do have a system. It's an adverse event reporting that's mandated using a structured taxonomy. And it's called the Common Toxicity Criteria for Adverse Events. But this is typically reported by research staff because even physicians cannot handle all the sort of minutia. It goes on a scale shown in the box there for rash from 0 to 5. 0 means you don't have any rash. 1 means a rash that's not so bad. Grade 2, minor rash. Anything that's grade 3 or 4 is something that's going to require a modification. Typically grade 4 is going to be a rash bad enough to put you in the hospital. And a grade 5 rash, grade 5 anything is death. So it's a rash that's so severe that it led to like a severe burn. It leads to to patient death. So we use that. When you pick up any drug, any cancer drug, you will see this grading system for symptom severity. But what happens in routine care? In routine care, clinicians just listen to their patients. They voluntarily report, totally voluntary, unusual adverse events, which then get acted upon or ignored. Yeah.
So it seems like there's a chicken and egg problem here, which is that on this inventory, you can only list the symptoms that you know to look for.
Yep.
So, like, how do new symptoms get added to that if they don't exist and you have no mechanism for even getting the reports?
Yeah. So when people do clinical trials, they have what they call anticipated symptoms and unanticipated symptoms. So anticipated symptoms are things that you are worried about based on what the drug did to guinea pigs and rabbits and mice, and you know you're worried about rash and headache and blindness and a whole bunch of things, and so you're systematically going to ask about all those things. But you're not going to ask about loss of taste, right, or hearing loss, right, because it's not something that you anticipated. If you start noticing it, if patients report it to you, you're in a clinical trial, you're supposed to record it systematically and score it and say, gee, all my patients on this drug complained of hearing loss. new onset blindness. You're supposed to report it and grade it for attribution, and there's a whole big symptom library, but how often that actually happens in clinical trials is suspect. So we're really good when we anticipate. When we don't anticipate, it's more haphazard. When you scale out to the real world, it's extremely haphazard. Does that?
Yeah, and it
Does that
seems
make
like
sense?
the problem.
And we miss stuff.
Because it's not even clear the patients know to say anything, right? So I'm getting cancer treatment and suddenly I discover that I have, foot cramps. It doesn't necessarily occur to me that they're connected. And so it seems like there's a reporting problem just from the patient side too.
Absolutely. Yep. I mean, do we necessarily want to know about every single foot cramp and be drowned in like things that are false signals? So we've got a signal to noise problem. On the other hand,
Sometimes they are actually.
sometimes they do turn out to be really meaningful and actionable, and they do turn out to be hooks where we can alleviate suffering. And we do not want to destroy people. There are drugs that destroy people's hearing, and if caught early, we could have done something about it. So you're right, it's a balancing act. Other questions?
Just a brief question. That patient that you detected had low calcium,
Yep,
then it turned out that he had low magnesium.
yep.
Are the two related?
Yes, they're related. Yes, the body's system for resorbing calcium and magnesium is in the kidney, and the drug is a monoclonal antibody that attacks something called the EGFR receptor, and the EGFR receptors are overexpressed on the tumor, but lo and behold, it turns out that they're also expressed in a particular corner of the kidney. Those of you who remember biology, they're overexpressed in the ascending limb of the loop of Henle. So the drug glommed on there and prevented the body from resorbing calcium and magnesium, messed up homeostasis, made our patients sick. Very easy to fix. You bypass the kidney and give the magnesium intravenously. If you have them swallow magnesium, doesn't work because the kidney can't hold it. So we figured it out, right? But we should have figured this out through better data science. We shouldn't have had to wait so long, just as an example. Question?
So just back to Margot's question, I mean, what if people could put in anything that struck them, and you didn't have to look at it until there were enough reports that it seemed like it was important, and you could actually give feedback to people of what mattered to them? That is, you could use computer systems to do that aspect of things as well, and just give reporting tools to the patients.
So that's what I'm going to talk about now.
She didn't give me it.
So, yeah, that's what we tried to do. And I described this case sort of as a raison d'etre for this. So this is the learning healthcare system, Margot, I think, as you're saying. Is it cyclical? Is it a cycle? Absolutely. But ideally, we have to know what questions to ask. We have to assemble the data. We have to analyze it. We have to interpret it. We have to act on the feedback, and then we have to change our care and change what we do and keep doing this in sort of an iterative, cyclical process. And I'm going to skip through the details of this, but suffice it to say that there are lots of breakdown steps, particularly in medicine, which is a very complicated endeavor, and we have breakdown at every step. And in particular, we have a lot of breakdowns when it comes to data acquisition, curation, integration, and analysis. And this leads to immense missed opportunities, waste, harm, and really delay in discovery. So I animated this one. Bear with me for the animation, but I think it's kind of cute. So the standard approach to symptom monitoring is you have a patient, shown by the little guy over there on the left, and he's at home, and his clinicians, it's not usually one doctor anymore, that model's outdated, it's usually a team, are working in some healthcare setting. And the traditional model, the patient has to go to some healthcare clinic or facility. And he's got some hurdles to get over, which are basically that there's limited time and he may forget to discuss his particular symptoms, because it turns out people don't want to, a lot of people don't want to complain too much to the doctor. They have 10 things, they don't get to all of them, so they don't mention many symptoms. Or the patient perhaps is not gone to the clinic, but the patient is at home and doesn't want to pick up the phone to call the physician, or does pick up the phone to call the physician and gets put on hold or gets the typical runaround that unfortunately we've all had, even we experience it. So upending that paradigm is a less reactive, more proactive model where you actually ask patients to report their symptoms systematically. So again, the traditional paradigm is reactive, but the alternative paradigm sends the patient a reminder and says, please tell us how you're doing. And again, it's please tell us how you're doing in these 10 domains. Do you have a headache? Do you have nausea? Is your hearing any change in here? You can ask structured questions, and you can also ask, is there anything you want to tell us? Sort of just, and anything that we should know about how you're doing. That information gets stored back in a computer. Alerts then get sent to the clinical team who get structured reports. This brings the clinical team and the patient closer together. It's less reactive, it's more interactive in real time, and there's an opportunity in this more proactive report to actually improve care. So I'm going to skip through lots of research and papers and I won't belabor the point, but suffice it to say that there is abundant evidence that clinicians are unaware of up to half of patient symptoms. Systematic symptom monitoring has been shown to close this gap. A lot of foundational pilot work shows that patients, even low literacy, patients from low literacy, relatively uneducated, relatively computer naive, are perfectly willing and able to self-report, and it's easy to do. What's also important is that clinicians actually trust and believe patient-reported information. So we wanted to test the study hypothesis that proactive symptom reporting during chemotherapy would improve symptom management, leading to better clinical outcomes. Would any drug company let us touch their study or integrate alongside their study? Do you think? Absolutely, they would not, right? Because they're trying to get drug approval. So what did we do? I was working at the time in a very large cancer center, Memorial Sloan Kettering in New York City, and we picked patients not in a clinical trial who were getting chemotherapy for advanced cancers. We randomized these patients to two groups. The standard symptom reporting, which is that reactive, passive, if your clinician figures it out and picks it up model, to an intervention arm where the patients were asked to self-report 12 common symptoms, write in or add in anything else, like any stray things that they thought, prior to and between visits by either web or phone. And they got little prompts and reminders to do so. And alerts when they had severe symptoms went to nurses. These were sent to emails, and oncologists received these reports at visits. Now, I can tell you, do you think oncologists liked this idea or didn't like this idea? Any guesses as to whether cancer doctors wanted this or didn't want this? Any guesses?
I have a question.
Yeah.
Did you give them summaries or they had to look at all the details?
We actually gave them the details. We gave them the raw data. Perhaps not as elegantly as we could have. Yep.
I would think they would want it because at the time of the visit, number one, they're time limited, and number two, the patient is often not a good reporter, doesn't remember what happened last week or the week before.
Right. So they wanted it, but it's only valuable to them if it's integrated into their workflow. Okay? If it's some other place that they have to go look at, just like, if you got to go log on to some other website, it's a pain. If it's, behind some other firewall or curtain and requires another login, it might as well not exist. And of course, in a research context, you can't start by integrating something directly into research data can't be directly integrated into an electronic health record. So there's sort of like a research launch paradox, if you wish. So people were happy to have it at the point of care, but when didn't people want it? It's Thanksgiving weekend. I don't want to know that my patient has a terrible headache. Let's just wait till I see them on Monday. What am I going to do with my patient's bad headache Saturday night? I don't want to know. So on the other hand, there's a social desirability. I should want to know. I shouldn't want my patient to be suffering over Thanksgiving weekend if I was a good doctor. So we had a lot of social desirability bias. Physicians recognize that the healthcare system needs to be more proactive and less reactive, and that theoretically it's a good idea, but at the same time, their ambivalence stems from the fact that I already have too much email and phone calls, and now I've got to manage like all my patients' Twitter feeds for, you know, whatever systems are, for all their symptoms and every headache and every gas pain. So clinicians had immense ambivalence about this kind of research. So we decided to look at how patient-reported symptom monitoring would influence outcomes, specifically quality of life, emergency room visits, and survival. And we created a patient self-reporting interface, and I'm just showing you a couple of screenshots. We did lots of usability testing. If this sort of user interface looks familiar to anyone, this study was conducted in New York City, and it was launched about a decade ago. And this is what the Citibank, which is the dominant bank, this is the font and look and feel of Citibank login when you go to their ATMs. So when we did UI testing, all patients were familiar with that font. They liked that color scheme, which is actually shades of blue. That's what they wanted. So people were like, it looks like Citibank. Yeah, it looks like Citibank, because that's what patients were comfortable with and they could read. And not, you know, these are sick people, old people, people with bad vision, people who are not computer experienced, but everyone can take money out of the ATM, and so they could do this. So that's a pain example. We had it for shortness of breath. This is just an example. Patients click. When they click on something, it lights up. Yep.
I'm just curious, do you find that there's an age bias with the reporting? Are you getting more comprehensive reports from younger patients who are sort of more comfortable using the technology, or?
So it's really interesting. That's what we thought, but it's not true. It didn't happen. So we actually, when we did our randomization, we enriched for what we called computer naive individuals, which we defined as people who didn't have a computer at home or a smartphone at home. So because we wanted to really prove in this that you didn't have to be like a millennial with a smartphone. Because remember, this is a disease of 70-year-olds, right? Actually, once taught, once we overcame the activation hurdle, so it took us longer, we timed how long it took to train. It took longer to train older people, but once we trained them, they were more compliant. Actually, younger patients are less likely to continue reporting. They tend to extinguish and drop off more. So, not exactly. Did I answer your question?
How often are they reporting? Once a week?
So in this study, we asked once a week. In other studies, we have all kinds of studies. We tend to tag that to the severity of the condition. So for example, in bone marrow transplant, where patients are super sick, we'll do it once a day. If it's a twice a week chemotherapy, we'll do it every other day. So this was an every other week chemotherapy or every second week chemotherapy, so we did it weekly. But again, we set that based on sort of the clinical parameters. This is a mix of patients, so this is patients with advanced cancers, not leukemias, not bone marrow transplant, not leukemias in this study. So emails went to the clinical nurse, and this, to your question, they're really pretty ugly, but we just gave the clinicians, here you go, here's the data. It was an experiment, do whatever you want with it. No directives, like your patient had terrible diarrhea or they had no diarrhea. Here you go. And the clinicians could copy paste this into their note. Ugly interface, had to go to a different site. They could copy it and paste it into the electronic health record, or they could not look at it and ignore it. They weren't penalized or rewarded for doing it. And in a lot of experiments, patients are incentivized or paid to do these kind of things. That has no sustainability. Patients were not compensated. Physicians were. This was just implemented as though to see if it's something that could work in routine care. So, our primary outcome when we designed the study was quality of life, which we measured using really the standard validated index, and it was a well-powered study. We also looked as a secondary outcome at overall survival. And we enrolled and randomized 766 patients over about a 4-year period. And we closed our overall survival analysis in June 2016 with about a 7-year follow-up. And by that point, 67% of the cohort was deceased from their cancers. So what did we learn from the study? Patients self-reported their symptoms 73% of the time when they were prompted to do so. Nurses took action in response to the alerts 77% of the time when those alerts hit grade 3 and 4. They did not respond to alerts like grade 1 and 2. That could wait to the next doctor visit. So that was their threshold, and that's what they were instructed to do. We found high rates of reporting among both computer experienced and inexperienced patients. Oops. Something just happened with mine. Is that me? I might need a little AV tech. I'm not sure. Something seems to have just happened.
Just say, don't say.
I don't know, maybe you can. All right, I can describe what happened next. So what we found is that asking patients to self-report their symptoms kept people out of the emergency room, improved quality of life, and actually, to our surprise, was associated with a statistically significant improvement in overall survival. It actually helped patients live longer. What's the mechanism? Well, if you identify, so I'll just give you one specific example. If you identify a symptom like dehydration early, I'll just continue now, I'll catch up with the slides in a sec. If you identify a symptom like dehydration early, you can call a patient in and do something very cheap and inexpensive, like give them some salt water, keep them out of the ER, keep them from going into renal failure, keep them on time with their chemotherapy schedule, enable them to get more treatment. So it's not a shock. But again, this is a lot simpler. This intervention of just measuring people's symptoms is a lot simpler than a toxic drug. So we had this really nice proof of concept in almost 800 patients, with some other ancillary studies, but in one large major cancer center. That's not good enough in cancer medicine. That's not good enough for implementation. So the next step is we got a very large contract from the National Cancer Institute to try to develop a system to do this to scale. And the National Cancer Institute said, okay, you did this at Memorial Sloan Kettering, but you're replete with resources. It's like doing it at Harvard. Lots of resources, really engaged, smart nurses, special physicians. This is not Main Street. We want you to do this and integrate into clinical trials. And we want you to develop a system that will work in anywhere USA or anywhere North America. So they gave us a contract to do that. So first we had to go through the entire symptom library of symptoms that patients can report and figure out which symptoms are best reported by patients versus doctors. So patients are not good reporters of hypomagnesemia, low platelet count, and anemia. That's not what but they're good reporters of fatigue, nausea, vomiting, ringing in the ears, numbness and tingling in the feet, funny taste, right? So we went through each of these. So there are 89 of them, 89 symptoms that patients are better able to report. This is in cancer medicine, but they're pretty much the same symptoms in all of cancer medicine. What was our next problem? So we had these 89 symptoms, we had a taxonomy, but we had to figure out valid data structures and ways to capture this. One very specific example, cancer medicine often calls a condition called neuropathy. Does anyone know what neuropathy is? It's death of like the nerve endings. Diabetics get it in their hands and toes. Most patients do not know what neuropathy is, and they don't come in and say, gee doc, I have neuropathy. They describe it in different ways. A funny sensation, I can't feel my fingers, okay? So we had to figure out whether we were gonna have one word for it, 10 different words for it, because if you're eliciting symptoms, you have to be sure that the words that you're using mean the same thing. We call this construct validity. I'm sure you guys use a similar term. But we had to evaluate construct validity around the country, because people use different words in the South, the North, the East, and the West. So mind-numbingly painful studies to figure out how patients describe neuropathy around the country and then around the world. Neuropathy turned into numbness and tingling. The medical word that we use is pruritus. Don't ask me why. It means itching. Pruritus turns into itchiness. Right? Okay, so we had to do sort of this translation and validation and really develop a taxonomy to figure out a library. Then we went out to clinical trials, industry-sponsored, government-sponsored, and said, we have a system for eliciting patient-reported outcomes in clinical trials. Give us the 15 symptoms that you anticipate and a mechanism for, we essentially call it free write-ins, right? Free write-in comments as well. And we'll add on this patient-reported outcomes system to your clinical trial. Did any drug companies want us to do this? No, they did not. Because do you think that clinical trials underreport or overreport symptoms? Any hunch there? Yeah, they underreport. So what we did is, it turns out I did this with a team of people. This is my work alone, it's all teamwork. But I am the PI of a colorectal cancer trial that's accruing at 300 sites across the US, Canada, and Switzerland. And it's a trial of an old treatment versus, it's a sequencing trial. It doesn't involve a new drug. So no industry sponsor was gonna get upset. And it compares treating rectal cancer with chemotherapy or radiation. Okay, so two, but nothing that's on the docket for FDA approval. And we integrated this system into that clinical trial. So we have patients getting two very different treatments. Radiation, which causes terrible diarrhea, and the chemotherapy, which causes terrible neuropathy. And we asked the question, will patients report in a clinical trial, not at a fancy cancer center, but all over the country? And if they will report, will we be able to pick up the distinction in those symptoms? In the symptom profiles. Does that make sense? All right, so now let me show you what we found. I will catch up on the slides. Thank you for the technical. I'm just gonna skip. So these are the two systems. We developed a system, PRO-CTCAE, which is a companion to the system that the FDA mandates that clinicians use. So it'd be like, an example, professors grade students at Harvard on ABCD, and now you develop a system for students to grade professors that maps exactly to the same ABCD. That's essentially what we did. We just translated the terminology so that patients could understand it. The system consists of both an item library of these specific symptoms that you can choose from and software. Theoretically, could you include all 78? Theoretically, you could, but you don't want to burden your patients with reporting 78 different symptoms. They'll extinguish and they won't do it. And we've learned that really 20 is about the maximum. And when people do it on their phones, which we've migrated to, just clicking, 1, 2, 3, 4, 5, 1, 2, 3, 4 on phones, it's actually, we like people to stick to 12 or fewer items. So when you ask clinicians are reductionists. They just write, how bad's the person's nausea? How bad's their vomiting? How bad's their diarrhea? How bad's their neuropathy? They can do it on a scale of 0 to 5. But when we did the psychometric validations, patients are smarter than that. They're more nuanced than that. They rate symptoms along different domains. It's not just how bad is it. They're like, that doc, I can't answer that question. It's how severe is it? How frequent is it? And how much does it interfere with my activities? That's much more meaningful to patients. So although we wanted a nice, perfect one-to-one correspondence, we quickly learned that we couldn't, and we had to have a system that incorporated what matters to patients, which is that nuance and distinction. So we did a lot of cognitive interviewing to really try to get it right. So we ask patients to think back over the past 7 days, and they're depending on the symptom, they have domains of frequency, severity, interference. A couple have amount or simply presence or absence, and they all have consistent formats. I'm going to skip through the psychometric validation study. So this was the big randomized clinical trial. It shows at the top, this is the study schema. The patients in the control arm get 5-FU radiation and operation and then chemotherapy. And patients in the intervention arm get a tailored approach where they all get chemotherapy first. So this is a randomized trial that's actually still ongoing. It's nearing the finish line. It's recruited 915 of 1,120 participants, and it should finish in the next year. So this is typical of big cancer trials, and this is what you read when you pick up the New England Journal of Medicine. So we sent all these patients who were, I think right now we have 172 sites across the United States. We sent them a weekly telephone call by text to their phones or an email, and we asked them to complete these symptom reports. For external validity, we also asked them to complete quality of life measures, and we focused on specific domains that relate to the treatment of rectal cancer. And in the meantime, we had the patients and the clinicians report the exact same symptoms. So these are data for the first 915 patients. So just a little bit about who these participants are. The median age of clinical trial participants is 56. The median age of the disease is 71. So right here we have a generalizability problem, okay? Whatever the results of our trial doesn't really help you manage this particular cancer in the general population. We had a lot more men. Some of that's just reflective of rectal cancer being more common in men. The way we did this is on the due day of each week, patients got these automated emails or calls. They got up to 2 reminders. And if patients didn't self-report, we did have a backup telephone call just as a reminder. The actually IRB wanted us to do that to make sure that patients were safe. So our first aim was just to see whether this was feasible in a multi-center setting, and all sites were able to implement it with a central coordinator. And we did even better than we did at the major cancer center, with 93% of patients completing all expected data at all expected time points, 78% unprompted, and another 15% were retrieved with prompts. Compliance, adherence to reporting was similar, whether patients did it by web or through an interactive voice response system. Patients could choose at the outset. So the next question is, all right, if the collection of these types of data are feasible, are the data useful? Because there's a lot of concern with it's extended now to wearables and collection of this kind of data, that we're just churning up a lot of noise and that we're not really generating useful information. So again, we chose to implement this in a study where we already knew in advance that there are expected clear toxicity differences between the 2 study arms, just a different symptom profile. So on the left in gray are the symptoms as reported by the physician, and the horizontal bars go from 0, meaning none. The darker gray means more severe, grade 4. So what you can see here is that in arm B of the study, shown at the top, the doctors thought that the patients had more neuropathy, and that overall there's very little severe neuropathy. Grade 4 neuropathy would be neuropathy so bad that the doctors stopped the drug. Conversely, you can see there's more diarrhea in arm A and less than in arm B, according to the perspective of the physician. So this is what the physicians had to say. So then we look at what the patients have to say, and that's shown in blue. Now, first of all, we asked patients, we did the psychometrics, to get at neuropathy, we had to ask patients about both severity and interference. So 2 constructs, that's why there's 2 ratings. So the first thing you can see is that the patients agree with the physicians that neuropathy is worse in arm B, right? So the directionality is similar. They both think there's more neuropathy in arm B. But overall, patients report a lot more symptom burden than the patients. So by restricting ourselves to clinician burden, we are missing symptoms. Any questions there? Diarrhea, again, flips the other arm, flips the other way, not quite as much difference. Patients thought there was bad diarrhea pretty much in both arms. And again, this was done for 12 symptoms. I'm not going to belabor and show you all of them, but pretty much this pattern, this cord, electric problem, I'm not sure. This pattern pretty much extended. So we did one more experiment. When you do a randomized trial and you have over 1,000 patients, you can do a randomization within a randomization. Does that make sense? Mm-hmm. So what we did is we took half the sites, because remember we have over 150 different clinical sites, Kansas, Nebraska, Montana, California, we're all across the country, and every province in Canada. And at half the sites, the physicians saw what the patients reported. And in the other half, they didn't, which gave us an opportunity to ask the question, is there convergence, right? Are doctors going to stick to their scores, or are they going to look at what their patients report and say, wow, if my patient says their diarrhea and neuropathy is bad, I'm going to believe them and I'm going to up my own score. Does that make sense? Any questions on that? So we asked that as a corollary question. These are what the tables look like in toxicity. I'm going to skip this. This just shows reporting week by week that patients continue to do it, that they don't extinguish. This compares the two arms. So here we did a cluster randomized trial. Inside the trial of parallel reporting sites where clinicians didn't see their patients' reports, or shared reporting where they did. Okay? And the way we operationalized this was a very simple standardized adverse event reporting form. And if you think we're in the modern age, we're not. This is what case report forms look like in clinical trials going on right now. These are parallel, not shared clinician report forms. The patient scores aren't there. The doctors just look down these list of symptoms and score how severe the patient symptom burden was. And this is the standard of care. This is the way symptom burden reporting is done. The shared reporting, same information is on the form, but we stuck the patient reports right in the middle, so it was right there, so when the clinician scored it, they had the opportunity to modify their score. No one was paid, no one was nudged, no one was reminded. We just sort of wanted to do this experiment to see what would happen. Our hypothesis is that clinicians will modify their reports when presented with the patient information. In other words, we hypothesize that doctors actually will believe their patients and don't think that their patients are lying to them about their symptoms, and that we would see convergence between patient and clinician reports in the shared group, but not in the parallel groups. And we also hypothesized the corollary, which I think makes it a stronger experimental design, that we would see no difference in patients' reports between the two groups, that the patients themselves shouldn't be reporting anything different.
Did the patients know that the doctors were going to see their scores?
They did. They were not blinded to that. Yeah. The consent form for the patients, basically said that, so we really didn't have a way of doing it. Suffice it to say that clinicians modified their grades when exposed to the patient reports. There wasn't absolute convergence, so even with shared reporting, patients report more intensive symptom burdens. So there's partial convergence, but not complete. Does that make sense? So it changed it, but PRO CTC reports continue to reflect a higher symptom burden. But this really gave us the opportunity to understand integration of patient-reported outcomes. So I just want to let you know kind of where this is going and what's coming next. This is all active reporting, right? But we also have an opportunity to get data now from passive reporting and biometrics, right? And we have a lot of work done and a lot of work underway, a lot of clinical trials, where we're attaching our patients essentially to Fitbits and other biosensors and trackers and monitors to try to not acquire data from them simply actively, but also passively. Now, we are besieged every day with the latest gadget gizmo maker who wants to come with his or her gadget or gizmo or startup company to measure something. Food intake, sleep quality, sex life, everything. And each one is an app. Each one is a standalone app. Each one is investigational, so it can't go inside the electronic health record. And each one, to be actionable, requires our busy eyeballs to go look somewhere else. Does it, right? It doesn't work very well, so we are facing a massive data integration problem. And we have this paradox of, in order to really do a good test of something, it has to be inside the system to be tested. On the other hand, we can't put things inside the system until they're standardized and approved. Again, it's a sort of a chicken and the egg problem. So we're really trying to figure, and we're also a little bit limited by technology, because we would like to monitor our patients' vital signs for temperature, for example. We have most of our work is with activity sensors, which are working the best. We have some with temperature sensors are not working very well. They go lots of false alarms. In cancer medicine, if patients have fevers, it can be a life-threatening infection. And if we're calling people for false fevers, again, we create a lot of false alarm, a lot of excess labor, a lot of worry. We send a lot of people to get blood tests that they don't need. So again, this technology can go overboard. But the Fitbits have proven so far, we don't have elegant randomized trial data like I showed you, but we have nice preliminary data, which I won't show you in the interest of time, where we can track who's not getting out of bed. And when patients stop getting out of bed, we call this ecological momentary assessments. That's the lingo some of you may know. But patients, a lot of our patients go to the mailbox every day, walk their dog 3 times a day. We can see the dog walk. We can see the trip to the mailbox. We can see exactly what's happening. And then we can see when it doesn't happen. And we can see people's patterns. They get chemotherapy every 2 weeks, and for the first 4 days after their chemotherapy, they only walk the dog once a day, and then they go back to walking the dog twice a day. We can see these patterns. We have amazing data from these patterns. And when the pattern is disrupted, we can call, right? We can call them and say, hey, what's wrong? You haven't gotten out of bed, right? Have we done it with nutrition yet? No, because the technology is not quite there. Temperatures, we've tried it. The technology's bad and it's not working. So there are lots of glitches. So now our next strategy is to integrate active patient reporting with passive ecological momentary assessment. So why is this important for computational scientists? The healthcare system does not have the capacity or skill set to respond to all this data. We just don't. Moreover, we face a huge challenge because we've built all this data, and the number one question I get when I present this kind of data on the healthcare side is, this is great. This improves the value of healthcare delivery. We can see how this could cut healthcare costs, help people feel better. What's not to like? But who's going to monitor the data? Who's monitoring the patient reports Thanksgiving weekend and calling the patients with bad headache? Who's on call for the data? Right? So we take turns as physicians being on call and covering emergencies. We've all had a relative who called the doctor on call Saturday night, and right? But who's on call to monitor the data? And our current system, we need a human on call to respond and say, gee, you've been vomiting, you've had terrible vomiting, you probably need to come in, and what we need great data scientists and great computationalists is to figure out how to give patients appropriately tailored self-help. Patient self-help right now is going to CVS or Walgreens. They don't want to call. They go to WebMD. They look online and they get generic help. But that is a disaster for cancer patients because our patients are already on 17 other medications and they have polypharmacy and it's going to interact. They need algorithms that tell them what medicines they should take conditioned upon the medicines that they're already on and what they have taken. So when those algorithms run off not only the reports and the passive biometric data, but also on what's in the electronic health record that they've been prescribed, or coming soon, FDA approved in the last, very recently, digitized pills so you know not only what they've been prescribed, but what they've actually swallowed. Now algorithms can start to work. So again, one solution that we're looking at in the medical field is to develop a new class of health professional, essentially data concierge, because physicians don't want to do this. We do not want to be looking at screens and monitoring people's symptoms 24/7. And we're probably too highly paid for it to be a viable model. Nurses could do it, nurses are doing it. It's kind of been a nurse-centric activity, but there's some trepidation and anxiety that maybe this isn't the best use of nurse time either, that really we need algorithms to help us solve this problem, except maybe in extreme cases. But we need the algorithms to figure out which patients need a phone call and which can be pushed the appropriate self-help data that is truly personalized and tailored. So let me stop there and take some questions.
So we have time for just a couple of questions.
So I know that Partners has invested a great deal of money in a medical record system that causes hospitals. Does much patient-reported information get into the clinical record in the real world? And sort of a follow-up to that is, that's a very large data set that presumably has some utility for sort of data mining and machine analysis. Is there much of that or enough of that happening now?
So it's a big question. So the electronic health record that Partners, which includes Dana-Farber, it includes all the Harvard teaching hospitals with the exception of Beth Israel Deaconess, is called EPIC. And EPIC is one of about 3 dominant EHRs in the nation. For cancer medicine and for the cancer hospitals in the nation, it is the dominant EHRs. So I would say we are tremendously engaged in data mining and data integration. There's some major obstacles. Some of those obstacles include the fact that EPIC is a privately held company, that it's not open source software, that it's very difficult to, Margot's smiling, it's very difficult to change and add it. So essentially what we have to do is basically build applications that float on top of it. We have to go to Wisconsin and become EPIC builders ourselves. And essentially we kind of end up working for the company in the sense that we're a vanguard practice and we're sort of innovating at the cutting edge. So we develop new strategies and then they take them and then they build them. But there's a delay. It takes them 4 or 5 years to do a build, and they're less interested in building something for cancer medicine. Their primary focus is to build something that works for cancer medicine and cardiology and that works for everything. So the way we've handled that is we're really focused on developing data standards and taxonomy, because that's the biggest thing that's missing, and that's what companies do a terrible job at. It's also a critical obstacle for machine learning. So I can't tell you how many PhD thesis projects I've seen where people say, I want to mine Partners' data to look for neuropathy and see who has bad neuropathy. I've got great NLP chops, machine learning chops, AI chops, like I can just go, like, here's all the EHR data, and the word neuropathy is sprinkled everywhere. But there is no, people need a data standard. What's meaningful neuropathy? What's nonsense neuropathy? We use a lot of words in medicine that are really confusing for NLP algorithms. Can't rule out neuropathy, right? We have issues of attribution. So machine learning and AI are really important, and they're incredibly important, but they're really stymied by both structural issues related to sort of the architecture of the EHR, the regulation of the EHR, but they're also stymied by us, the clinical community. Once we have developed data standards, this is trivial neuropathy, this is meaningful neuropathy, this is important, then you can have a go at it and deploy AI and machine learning. So our emphasis as clinicians and where we see our unique role is to really work on the data standards and the metadata and the validation and the psychometrics, which will then make it possible for computational scientists to really apply state-of-the-art techniques. But it's much harder when people just say, okay, give me the data, and they apply techniques and they generate these algorithms, and we're like, okay, you generate an algorithm, but I can't use it. It's not helpful or meaningful or useful. So again, I think one of the reasons why I'm here is that it's going to be better all around for patients, for clinicians, for cancer researchers, I think, and for computational scientists of all flavors, if there's a little bit more partnership and understanding of the constraints on both sides. Does that answer your question?
I meant that we have to give up the
Sorry.
room.